Our second section today is measurement analysis. I've got a few vocabulary terms here for you. If I do go too quickly, remember that you can always pause the video if you need more time. The first term, parallax, is the apparent shift in the position of an object when it is viewed from various angles. So, for example, you may have, in chemistry, measured water inside of a cylinder. And when that happened, you might have noticed that the water level dipped a little bit when it was in the cylinder. When you were taking your measurements, it mattered what your eye level position was. If you were to measure from up above, you might have viewed this mark as the level of measurement for your water inside of the cylinder. However, it's correct technique to get eye level with the cylinder. A little eyeball there for you. Okay. That way when you look across, you're reading the bottom of that curved part called the meniscus. This one would be the correct level to read. Another example you might try is to hold your thumb out at arm's length in front of you and look at it with your left eye closed and then with your right eye closed. And it'll look as though your thumb changes positions even though you're holding it still. So those are a couple of examples of parallax. So when we do our labs and you make your measurements, you want to make sure that you're using the proper technique and that you're getting eye level with anything that you're needing to take a data recording about. I included the next two terms in your notes, precision and accuracy, because sometimes people might get these confused or think that they're the same thing. They are in fact different. Precision is going to be the degree of exactness to which a measurement can be reproduced. So let's say you have a lot of trials in your lab experiment. If you get the same answer over and over again, that's going to indicate precision. So that reproduction of data is the key to recognizing it. Whereas accuracy describes how well the results of a measurement agree with an accepted value. You might get this on the first try. In order for something to be accurate, you do not have to have reproduced values. You just need to get the result that agrees with the expectation that you have in the lab itself. So only one trial is necessary for accuracy. To demonstrate the difference, I thought it would be helpful to use arrows on a bullseye target to represent the different cases. So we'll start with a bullseye that lands, an arrow that lands to the side, but I'm able to repeat that measurement. It may not necessarily be correct. I'm aiming for the bullseye and I don't hit it, but I am able to reproduce this measurement over and over again. So this would be an example of precision. Now let's say I had one dart and I were going to throw it at the bullseye, and I'm aiming for the center. If I throw my dart or aim my arrow and hit it on the bullseye on the very first try, this would indicate that I have accuracy. I also have two more examples. There might be a possibility where you have someone throwing and they are absolutely horrible and scatter arrows or darts all over the target. So let's say this is you. There's no grouping of our arrows and none of them hit the target. This is neither precise nor accurate. And then our last case would be if you're an excellent dart thrower. So let's say we're aiming for the bullseye and all three of our arrows hit it. This is both accurate and precise. So there are four possibilities. Precision is when you have a grouping that is not hitting the target you're aiming for. Accuracy is hitting the bullseye. Neither. All of our data is scattered all over. There's no grouping and nothing in the bullseye and the target that we're aiming for. And then lastly, we have both. We're really good. We were on target and we were able to reproduce those measurements. So that would be an example of both accuracy and precision. In our labs, we're going to be interested in how well we've been able to achieve the desired results. So we have something called relative or percent error. This is a formula that you're going to use throughout the year, so it's going to be good to keep it handy. We're going to say accepted value minus experimental value. And notice I have absolute value signs on the top. And then we divide by the accepted value and multiply by 100. Okay. Again, the accepted, this is what you're aiming for. The EXP is the experimental value. And this is what we actually achieve in our results in our lab. So again, the accepted is what you're hoping to get. The experimental is your actual data. The closer to 0% error or relative error you have, that means you are more accurate. Next, we have some sources of error. So there might be times that you don't achieve a low percent error. Something might go wrong. And if you ever have over 10% error in any of your labs, I'm going to ask you to come up with some possible sources of error and explain what might have gone wrong in your lab and how you might be able to fix them in the future. We're going to group our sources of error into 3 categories: personal, systematic, and random. And so what I did for each of these is just gave you a couple of examples. Personal error would be maybe carelessness in reading an instrument. So we talked about parallax earlier. Perhaps you did not get eye level and you recorded the wrong data. Another personal error might be recording observations. You could tell your partner that you got a length of 200 centimeters and they wrote down 200 meters. So maybe they had the wrong units, or they transposed some numbers, or left off a decimal, or something like that. You could also have a personal error in mathematical calculations. So if you're typing on your calculator and maybe you hit the plus sign instead of the minus, or something like that. Personal errors are like a one-time careless mistake. You just really weren't paying attention. On the other hand, we might have a systematic error that could be associated with a particular instrument or technique. So for example, if there's something wrong with your equipment, then that would be a systematic error. So you're using the triple beam balance and it's not zeroed properly, so you're getting the incorrect mass measurements. That would be a systematic error. For this part that says bias on the part of an observer, this would be something perhaps like a bad habit. It's not a one-time careless mistake. It's something that habitually is done over and over again that's incorrect in your lab technique. So for example, if a person always rounds up to the next whole number, regardless of whatever the actual data would be, that would be a bad habit. You want to record as much precision in your data as possible. Our third category for sources of error would be random. There might be times when you've gone back and you've checked your calculations, perhaps you redid the experiment, and you still can't figure out what's wrong. It very well may be a random error. You don't know why something is occurring and giving you bad results. Another example would be if you're in the middle of a lab and the fire alarm goes off. So you didn't know there was going to be a fire drill. That was unpredictable. You go outside, you come back, and let's say we were taking measurements on temperature. Well, obviously, if you had to take measurements that were every 30 seconds or so, you couldn't do that during the lab. So you're going to end up with some sort of error because of that unpredictable item. There also might be something that's beyond your control. So for example, if we're working with lenses and we need to focus some sunlight through our lens in order to look at those light rays, but it's really cloudy outside and we can't get the data we need because we need the sunlight to go through, that's out of your control. That's something that you can't help, so that might be an example of a random error. And lastly today, I want to talk a little bit more about some graphing methods. There are going to be some labs where you take data, you graph your data and look at the trends, and then you're going to make some predictions and then go test those predictions and see if you were correct. One thing that you might have to do is interpolate your data. So this would be an example of estimating values between known data points. So you're going to predict something between two values that you've already tried before. We also have extrapolation, which would be predicting values beyond the range of data points. Now for each of these, I wanted you to see if you could figure out which one is more reliable, interpolation or extrapolation. So let's look at an example. Let's say we've got some data points that are along our graph, and you were asked to interpolate. So you might take a guess and say, you know what, between these first two data points, I think that I can predict something that's going to happen right there. Okay, that seems pretty reasonable. Now let's say you're going to extrapolate. Beyond the range of the blue data points that we started with, I'm going to predict what's going to happen out here. And then you go back and test it. Well, which one of those do you think is more reliable? Hopefully you said interpolation, and I'll tell you why. Because with our data, we don't know if this was going to be some sort of linear trend, or it may have turned out to be an exponential trend, and we just didn't take enough data to see that pattern occur. So our actual result might have been somewhere out here. So interpolation, because you've got two data points on either side, it makes it much more easy to predict something that's going to give you more reliable results. So in conclusion, today's notes we talked about graphing, so how to plot a graph, also different graphical relationships. There were 4 of them. And then we finished up with some measurement analysis. We talked about parallax, accuracy and precision, and then some sources of error that might occur if you happen to have had a large % error. And then lastly, which is more reliable, interpolation or extrapolation. So I hope that summarized well for you, and you'll be ready for the next class.